Welcome to the 2023 edition of Trieste Next. I'm Luca, an AI-generated speaker who will be presenting our festival. In the next 3 days, we will be talking about a new world. This is the title of the 12th edition of our Festival of Science. Innovation and research, the ethical limits and new frontiers of science will be the center of all our events. What will the new world be like? Will we be able to face the challenges of the near future applying new scientific discoveries and technologies in a sustainable way? This will be the underlying theme of Trieste Next, with 100 events in English and Italian, the Science Book of the Year Literary Award, 300 speakers, 70 activities for schools and 40 exhibition spaces that are open for everybody in the city center close to the town hall. Before leaving the floor to our next panelists, I would like to thank all the scientific institutions within the Trieste City of Knowledge Network, the main partner together with our content partners and sponsors for supporting our project. Mostly, thank you to the 200 volunteers who are helping us. I have one last remark for all of you. The next event will last 75 minutes. Our speakers will answer all of your questions in the final 15 minutes. Also, please remember to check the updated schedule of events on our website. Thank you and enjoy our festival.
Good morning, welcome, and thank you for joining us. Well, the right treatment for to the right patients at the right time. A revolution is coming to medicine, and right now we are going to explain why and how. Let's try. Thank you for joining us. It's a pleasure to stay here with you and with you, of course. As you know, Trieste Next is a platform for both amusing with science and learning. Trieste Next is a platform for networking with the scientists and exploring the new frontiers of science and technology. This panel is organized by Burlo Garofolo University of Trieste, and this panel is focused on healthcare innovation, is focused on innovative drugs and therapies, and so is focused on tailored medicine. As you know, many common medicines are not effective for many people because people don't respond to medical treatments the same way, so we need a new approach. Doctors have to treat people, also people with the same disease, with using personalized plans. In other words, as I said, the right treatment to the right patients at the right time. Well, we are in the middle of a new era of medicine, and right now we are going to talk about the challenge of personalized caring. With our panelists, of course, Alberto Tommasini, doctor from Burlo Garofolo-Institute for Maternal and Child Health, professor of Pathology at University of Trieste. Thank you for joining us. Welcome. Oscar Della Pasqua, professor of Clinical Pharmacology and Therapeutics from University College London. Welcome. Marianna Lucafò, assistant professor of Pharmacology from University of Trieste. Thank you for joining us. And Paolo Vicini, Chief Development Officer, Confo Therapeutics. Welcome, please. What is personalized medicine? What can tailored medicine do for us? How does tailored medicine work? Well, there are the main questions. And Paolo Vicini, let's start with you. You are expert in pharmacology and drug development, so please explain us how medical centers and university and biocompanies are working to produce tailored therapies. So let's start from the R&D challenges, and please, of course, explain us why personalized medicine is important. Please.
Thank you very much for the introduction and for having me, and thanks everyone for being here, both those of you that are here in person and those online. My intent here is to give a brief overview of how new medicines are discovered and developed. And so I would like to take you today on this journey, which is truly a journey, right? It's a multi-year, multi-process journey that involves a lot of talents, and starts in the laboratory, where chemistry, for example, or biotechnology is used to find new molecules for therapeutic targets. And then it moves on to the clinic, to the hospital, where these new potential medicines are tested in patients and used eventually to treat them. And then to the pharmacy, where these approved medicines are then dispensed to the public. I know that some of us are going to talk about AI, and I play with AI with just some visuals. So what you have here are AI-generated laboratory, hospital, and pharmacy of the future, which I'm going to use here for illustrative purposes, right? But like I said, the journey goes through different places, but also through time. You know, we start by discovering new medicines against molecular targets that have the promise to be therapeutic. So the idea, the hypothesis is that if we develop a new molecule against a certain molecular target and we administer the molecule to people according to a certain schedule and to a certain dose, that will lead to improvement in disease, right? So that's the decadal, the 10-year effort in chemistry, where you sort of start developing molecules and it can last a long time, and of course it's expensive. And then out of a very large number of molecules, and I'll tell you how many later, only 200 will enter preclinical testing. Preclinical testing will last about 6 to 12 years, and then of these 200 molecules, only 5 will be tested in humans. And out of those 5, only 1 will eventually make its way to pharmacy shelves. So it's a high risk endeavor. I have to say it's a very fulfilling journey, because it's one of the most complex, I think, human adventures, to develop a new medicine, to actually be able to influence biology, to influence the disease state, and actually make patients feel better. So some of you may know about this process, some others not, but my intent here is to give a basic introduction as to how this works and really how much time and effort it takes to bring a new molecule to patients who ultimately will benefit, and why personalized medicine is important. I mentioned that at the end. So drug discovery is where this all starts. It starts, like I said, in the chemistry lab, but before that it starts with a biological hypothesis. There is a target, there is a molecular target that when it's hit or locked by a certain molecule will make the disease process improve. And so the idea is, what are these molecules that can change biology, that can bind to the molecular target and in turn change things for the whole organism. And that's how our chemists or biotechnologists spend the time finding these molecular keys that can act on the target lock, so to speak, a lock and key mechanism. And then how many of these compounds are discovered? 10,000, say, on average. And of those 10,000, only 250 will progress, and then the numbers 5 and 1 that I mentioned later. So it's a big funnel where there's a lot of attempts at the beginning, there's a lot of potential molecules that may work, but of this potential, only very few will actually make it through, right? And so the process that I mentioned earlier is a process of continuous testing, where we test these new drugs, these new potential medicines through various steps at the molecular scale, at the whole organism scale, preclinically, clinically, on and on and on, until at the end we're left with one medicine. And then, of course, the end of this all, I mean, like someone said, patients are waiting, and so the end of this whole process is the pharmacy, where these drugs, these medicines are now dispensed and dosed at the right amount with the right frequency, so that they can make a difference for patients in need. But the truth of the matter is that because of variation, some of it is genetic, some of it has to do with other factors that are different from genetics, but even when we take a medicine at the same dose as everyone else, at the same frequency, we will respond differently. So every person is different, and that has to be kept in mind when we develop these new medicines. And the growing discipline of personalized dosing intends to understand what makes us different and how can we tailor the dose and the frequency of the medicine so that it's optimal for every one of us. So I mentioned this briefly. Why is personalized medicine important? Because we're not the same. And it's interesting because you have a pill, it's a certain dose, a certain milligram amount, and you give it once a day, twice a day to a group of people, and none of these people will respond the same as anyone other. So the understanding of the factors that shape these differences between individuals is very important, right? So, and how do we gather that knowledge through the journey that I described in terms of developing a new medicine? We gather it through experiments or tests that involve a growing number of people from phase 1, which is essentially a safety study, less than 100 people, where we try and understand how the molecule moves through the body and whether it's safe, to phase 2, where we begin to study patients, the intended population that the molecule wants to treat. And then, of course, we gather safety information, identify side effects, measure efficacy, and the numbers go up. And then phase 3, where we essentially keep monitoring side effects, but with a particular eye to efficacy. We want to rigorously test statistically whether the molecule, the new medicine, is making a difference for patients given a certain measure of well-being. And as you see, the numbers go up from less than 100 to more, to 100 to 300 to 1,000 to 3,000. As these numbers grow, we gather an understanding of what makes everyone different for a certain molecule. Now remember, this journey is for one molecule. Every one of these molecules that are in the funnel will start the journey, but out of 10,000, only one will get to the end, because they will stumble at one point across the way. Most of these candidates, most of these potential medicines do not become medicines, and the reasons are very diverse. We don't understand the disease very well. It's too expensive to make. It's not safe. We give it to patients and nothing happens. There's low levels in the body. It doesn't work. IP, intellectual property, stands in the way. It's not selective for the intended molecular target. It has side effects that make it unsafe. It doesn't keep on the shelf. It's unstable. It goes bad quickly. Competition makes it impractical, and maybe the rationale in the biology is not as strong. So there's a number of reasons why this funnel gets narrower and narrower, and I listed some of these here. But one of the ways that we have to improve the efficacy of medicines is to understand the determinants of variability. What makes us the same, but what makes us different, right? So some of the challenges that I think the other speakers will talk about a lot more in detail are, do we know individual variation? We begin with studying hundreds of people, then we end studying thousands of people. Is that enough to understand variation? Does individualizing the dosing actually make a difference for anyone? Maybe the spectrum of activity of the molecule is so wide that specific dosing doesn't really make a difference from a therapeutic standpoint. Is it timely? Is it practical to individualize dosing? Is it too expensive? And especially, how do we train the medical professionals of this century so that they are prepared to personalize medicine when it's needed? So these are all challenges that exist on the background of this, what I think is a very exciting journey, and I hope that there'll be some questions from you at the end of the panel about this. But with that, I would like to hand this over to the next speaker so that they can talk about some of the specifics of this journey and what happens when you personalize those things. So thanks very much for listening.
Thank you. Thank you so much, Paolo Vicini, for this journey. You said the discovery of new drugs is a journey, not easy, of course, and you say we are all unique and everyone's genetic makeup is slightly different, and depending on our genome, we don't react the same way to many drugs. I read on the Lancet a study of 7,000 people. This study is published in February, and this study found people had not the same side effect when the doses of certain drugs were tailored to their DNA. But let's go on to speak about ideas and projects that can transform the future of healthcare. Let's go on to speak about the rise of tailored medicine, and let's try to explain how medical centers and universities around the world are working to come up with new information that will help doctors pair the right patients with the right medication. So the question is for you, Marianna Lucafò. I'd like to know your lab's activity and what about the role of stem cells and organoids. Please.
Thank you very much, and good morning to everyone, and it's a pleasure to be here. So, to promote personalized medicine, in our laboratory we need to have models which mimic the characteristics of each individual patients. As already introduced by Professor Vicini, there is a great inter-individual variability in drug response. If it doesn't work? Okay, yes, perfect, thank you. There is a great inter-individual variability in the way people respond to the drug. Generally, patients with the same disease take the same drug at the same dosage. And of course, most of the patients will respond to the therapy, so they will benefit from the therapy, but there is a percent of these patients will not respond to the therapy or will suffer side effects. Therefore, to reduce treatment failures, it's important to identify predictive biomarkers to guide the choice of the right drug for the right patients. The approaches used to identify biomarkers for predicting drug response and patient outcome are based on the use of biological samples derived from our patients, such as biopsies, so tissues, or blood samples, on which genomic analyses are performed. Basically, the patient's DNA is analyzed to look for an association between the presence of a variation in DNA sequence and the drug response. Sometimes this approach is not sufficient because the drug response is a complex phenotype where several factors are involved. The genetic, of course, is one of these variables, but it's not enough. In this context, a promising tool useful for predicting drug response is based on the use of patient-derived organoids. What is an organoid? An organoid is a three-dimensional culture systems, also known as mini-organs, as they mimic some of key multicellular, anatomical, and even functional characteristics of real organs. As you can see from these pictures, in our plate we can generate different kinds of organoids. We can generate intestinal organoids, liver organoids, cerebral organoids. Organoids can be derived from pluripotent stem cells or adult stem cells. What is the difference? Pluripotent stem cells are stem cells able to differentiate in all cell types of our body. Adult stem cells are not pluripotent. They are multipotent stem cells. They are resident stem cells, tissue resident stem cells. So we have found several types of adult stem cells in our body: gut, kidney, brain, blood vessels. So the adult stem cells can be differentiated into specialized cell types of that organ, of that tissues. How can we generate organoids starting from pluripotent stem cells? This is possible because we are using cellular reprogramming and induced pluripotent stem cells technologies. By using a reprogramming method, it's possible to go back somatic cells derived from our patient, I mean fibroblast blood cells. They could go back to pluripotent stages by forcing the expression of several, sorry, I don't know what is work, okay, so I don't know, that doesn't work. By forcing the expression of several proteins such as OCT4, SOX2, so these proteins are very important to go back the adult stem cells in pluripotent stem cells. Once patients induced pluripotent stem cells have been obtained, you can differentiate them into all the cell types, so in all these kind of organoids. Of course, to obtain these results, you need several weeks and months. We have another possibility. In this case, we can generate organoids starting from adult stem cells. This protocol is easier, is faster than the first one. Why? Because you need just a biopsy. You need just a tissue from which you can isolate adult stem cells, and in 1-2 weeks you can obtain your organoid. Of course, what is the limitation? The limitation is due to the accessibility of the tissue. It's easy to do, I don't know, for example, an intestinal organoid. It's more complicated to do a brain organoid starting from adult cell cells because I need biopsies derived from our brain. In our laboratory, we generate, for example, intestinal organoids starting from biopsies from our patients. As you can see, we need just 2-3 small pieces of tissue, and the first step is the crypt isolation. Why we need to use this crypt? Because the mammalian intestine is organized in 2 structures connected to each other, named crypts and villi. At the bottom of the crypts are localized the adult stem cells. So in this way, we can expose to these stem cells different growth factors, different stimulus to obtain intestinal organoids. Independently from the protocol used, organoids need to live within a supportive matrix. This is the reason why we need to create this like a drop, and the organoids can grow inside. As you can imagine, in the field of biomedicine, there are several applications based on organoid technology. For example, they are a great disease modeling. Why? Because they preserve the genetic and epigenetic background of our patients, for example. They are suitable for biobank. Why? Because you can expand and culture for a long time this culture system. You can frozen and thawed these cells all the time. And they, of course, are useful because you can use this model for high throughput drug screening. So at the same time, using just few organoids for well, you can test several types of drugs. Up to now, several papers which describe how the organoids derived from tumoral biopsies could be useful to predict the in vivo drug response. So if you generate organoids starting from tumoral biopsies and then you expose the organoids to different drugs, anti-cancer in this case, anti-cancer drugs, the response of the organoids will be the same of the patient. And this is a great result. Of course, all of these studies are observational studies. But the organoids can be useful also for other kinds of disease, such as inflammatory bowel disease. For example, in our laboratory, we are comparing the organoids derived from our patients with respect to the healthy controls to see the difference in terms of gene expression, proteins, to find a new therapeutic target. So they can be useful also to discover new drugs or to repurpose old drugs. And they are also useful to identify new molecular mechanisms for old drugs. We used, for example, this drug, azathioprine. The azathioprine is a drug used to maintain the remission in our patients affected by inflammatory bowel disease. First of all, we show that different organoids generated from 42 different patients highlighted a different sensitivity to the drug, and then it was possible to identify a new pathway involved in the mechanism of action of these immunosuppressive agents. So this drug, we know that this drug targets immune cells, but we hypothesized that also intestinal epithelial cells could be another cellular target. And it seems that the azathioprine can promote the remission of the disease also by increasing an important pathway named autophagy. This is an important cellular process. It's important for the function and the survival of the cells. During this process, the cells can destroy pathogens like bacteria. They can recycle damaged cellular part. So indicating that this is a new target for new drugs also in this disease. So of course there are several limitations and open questions about the use of these patient-derived cellular models, but we hope that in the future also patient-derived organoids can be used to promote personalized medicine. Thank you.
Thank you. So if I understood that the organoids are ideal in vitro tools, are useful tools, they serve as models, and they have potential application in drug development and tailored medicine, but there are a lot of questions with no answer. We need more research, of course, but the application of organoids are expected to expand in the future. Well, thank you, Marianna Lucafò. And next in line we have Oscar Della Pasqua, please. I think the healthcare ecosystem faces big and complex challenges, but at the same time is experiencing disruptive advances in life sciences and innovative technology. So what about the role of artificial intelligence in order to produce innovative therapies, tailored therapies?
Thanks for the invitation and for the opportunity to share some thoughts here about how we can personalize treatment, moving away from not only experimental data to disclosures. So I think what I'd like to give to you who might have very different background is evolution is always happening. It's happening all the time. And I think analogies help us understanding where we are going to here. If you think about the telephone, the first time the telephone was created, nobody thought that it would evolve to what it's now, a mobile phone. And perhaps we don't even know what mobile phones are going to look like in the next few years. So evolution is a fact that pertains all scientific areas. So what happened to medicine and to medicinal products in general? If we think about the way that we diagnose disease, physicians start with a medical examination, physical examination, and it evolved with lab tests, with data supporting decision making, up to the technologies we have today, such as PET scans that provide you support in imaging. Now, similar to monitoring of patients, we have evolved from the simple interaction with physicians to the support of technologies that evolved over time. And if we ask ourselves what happened to the way we are developing medicines, we saw an example of organoids, we saw an example of genomics and what is happening in the lab, but can we think a little bit further? So let's take a step back. 100 years ago, 120 years ago, this was the label of a product where you just sold a product with promises that you would cure disease, but no evidence. And over time, there was a framework, regulatory framework, that required us to start doing clinical trials, which was presented by Paolo Vicini. We have to evolve, develop data, generate data, and use that data to support our decision making. This was not more than 40, 50 years ago. Where are we going to now? And I think this is the point I'm trying to make here. Experiments are still the main determinant of how we make decisions. And what's happening today is probably that the complexity of the data that you're generating is taking over our ability to integrate and make decisions. So we need to basically realize that while on genetic level there's a lot of knowledge coming up, there's also understanding across this dimension that from cells you go to individuals and society. And the way we treat a patient is important on a personalized level, but from a healthcare perspective, we also need to make sure that treatments are effective and efficient. So we need to consider something like costs, for example. And how do we put these things together? We cannot carry on just generating data. We need to use something that is something beyond evidence. Data, I will refer to as evidence. We also need to make decisions about how a drug works in terms of benefit and risk using something we call inference. How do we integrate what we already know to take the next steps? So what I'm really proposing, and not myself only, but that's what's actually happening, is that big data is something everyone talks about, but what's happening today is also something called model. So we are talking nowadays about big models, mode-scale models, that convert data into a way that you can infer, you can make decisions from that data. And we are also now talking about, not only about experimental data, which we've seen an example of organoids for here, we are talking about synthetic data. How do I convert that experimental data into something that can be reutilized in a different context? We are also talking about not only real patients, we are now talking about virtual twins, patients that exist in the virtual space, so that I can explore how a treatment would perform. Now, why is this important? Think about airplanes. Everyone flies nowadays, but how do you build an aircraft? You don't put passengers and see whether it flies. You put that in a simulator, aerodynamic simulator, to see whether that prototype will perform well before you actually put real patients into an aircraft. We are trying to do exactly the same. Can I test a new drug, a new intervention in a virtual patient before I give it to the real patient, providing further confidence whether it's going to be efficacious and safe? So what we are looking at really is changing the paradigm from describing what we see, so descriptive evaluation or diagnostic evaluation, into predictive evaluation, predictive models, and of course prescribing recommendations that would then allow you to support medical decision and patient care. So how does it actually work? We have very limited time here to go through detailed examples, but what we are trying to show with modeling and simulation and other techniques such as AI, artificial intelligence, and machine learning is how you integrate information. So that information comes from clinical trials, from real world, from in vitro and in vivo experiments preclinically, and we make that integration through mathematics, through statistics, creating what we call in silico model. So we talked about in vitro model, we talk about in vivo models, in vivo experiments, but now we also talk about in silico models. And that in silico models then integrate virtual information that I can use, and we call that synthetic data, to explore the drug effects or the treatment response in virtual patients. And of course, how reliable, how trustworthy is this information? Now, for some therapeutic areas, we have established a very good correlation between what's happening in real life and what the models predict. So we talk about predictive performance, so I can trust the predictions from these models and actually use them to guide decision-making during drug development, during drug approval, and also in clinical practice. So we are now talking about not the drug has an effect, but the treatment has a certain performance, and I want to know all elements that come out of the treatment. So how do we apply this? So this is something we are doing in the context of regulatory approval and clinical practice, of course, but what we are really looking at, can we, first of all, use better the drugs we already have out there? So, and we talk about drug utilization, and one of the examples, I could give you a list of them, we have been doing in my group. We've been looking at how we look at personalized options. We call them model-based algorithms to inform pediatric dosing, to inform obesity, to inform underweight or overweight obese patients. How do you treat elderly? So you can look at what we call specific or special populations to start with, so that you can adjust the dose or adjust the regimen so that those individuals benefit equally from the treatment. And in certain cases, there are important changes in the way we dose those drugs. the focus till now has been quite a lot in pediatrics. Sometimes we look at obesity now becoming a problem across the world. How do you treat obese patients? How do you treat octogenarians? We now have individuals who reach 100 years of age. How do you treat those individuals, given that their organs are changing and they're functioning differently? So this is coming into the label of product. So this is just an example showing that you could use this drug without worry if there is another co-medication being used. So this use of model or in silico models is already coming into the label, into the prescribing information, and I could again give you lots of examples where it shows how in silico modeling is helping drug development and prescription. But as we said, we are trying to improve care as a scope of this session. So can we look at improved care in a broader context? So I will give you a very brief example, given the time we have here, to illustrate the concept, which is the patients are sick, they go to the doctor, and how do you treat a patient that comes to you once you know the disease and diagnosis? So the example I'm going to give you is in prostate hyperplasia. Is a disease where you have enlargement of your prostate, and one of the questions that we have been asking is, how do you treat a patient to prevent the disease from progressing? So the progressive enlargement of the prostate can be very traumatic for a patient in terms of complications. So can I predict what would happen? So we collect data from more than 10,000 patients, so again, big data, more than 2 million rows of data, just looking at clinical and demographic information, as well as phenotypic information. And what we did, we developed models in silico that would allow us then to understand the performance of the disease, so the progression of the disease and the treatment. And from there, we create virtual patients where I now look at the symptoms and the complications. So can I predict, given the characteristics of the patients, what will be the symptom trajectory, what happens over time, and the risk of complications? And the answer is yes, we can. So we do something we call clinical trial simulation or real-life simulation. I cannot go into the details, but it's basically a computer simulation, like you do with aerodynamics. You put the patient in a treatment in the computer, and you run that the treatment through a number of iterations. And I will illustrate, even though you might not be familiar with the area of prostate hyperplasia, IPSS is a symptom score, so it's a scale that shows how bad you are, and 20 is not a good score. I have symptoms. And what happens? I need to understand what will happen to this patient in a virtual setting. And one of the questions doctors can have is, should I give this patient a combination treatment, or should I give a monotherapy with a symptom, an alpha-adrenergic drug that might block some of the symptoms? Or should I go for a more efficient intervention? And you could also say, what if just hold on, wait, I will not treat you, so I can look at scenarios and decide how to treat this patient. So what we did, basically looking at the potential outcomes from this patient. If Mr. X or Mike, let's call him Mike, has a certain pattern, so we call them fast progressor, this is what's going to happen to his symptoms over time. If he's a moderate progressor, this is what's going to happen with the symptoms over time. The higher the score, the higher the burden, the worse the symptoms are, yeah? So that's what this line is showing. And you might have someone who is a slow progressor. So who is Mike? Is it a fast progressor, moderate progressor, or slow progressor? And we learn from the characteristics of these patients where he belongs. And we could find out that Mike was an average progressor, and his symptoms would behave like that if left untreated. However, if I give a treatment with monotherapy, which is a symptomatic one, yes, there is some improvement, but if I give him the combination, I will get even further improvement in the symptoms. And that's how I now start personalizing treatment using the information I have from Mike at the time I start the treatment. That could be genetic information, clinic information, or demographic information. And what we do, I look at risk. Okay, if I treat the patient with that particular symptom and signs at this moment, what will happen over time? And what we see, we have an app for that, but this is just a heat map showing what the risk will be over time, depending on the PSA, so prostate specific antigen, or the prostate volume of the individual at the starting point, or I could look at, again, the urinary flow. So I can look at different characteristics and assess what the risk of the patient will be over time for complications. So this is how we are moving now from experimental evidence to inference based on virtual twins and in silico data. Now, is this something that is just in science at university? No, it's something that's really resonating in the regulatory environment, in the clinical environment, where I would like to recall Alistair Breckenridge was the head of NICE, which is the Center of Excellence for Clinical Excellence in the UK. He usually said, science is evolving and regulation should follow science, and medical practice should follow science as well. What has happened in the past, it took a long time for medical practice to welcome and embed science in daily life and treatment of patients. I think we are now speeding that process, and hopefully this will be the future of our interventions in terms of personalized medicine. Thank you for your time, and we'll discuss later.
Thank you, Oscar Della Pasqua. You say evolution is happening all the time. You say medical practice should follow science. That sounds so interesting, amazing, but let's go on to see how to bring this evolution into clinical practice. And let's move to Alberto Tommasini for moving from the lab to the hospital. We say more effective therapies because tailored based on genetic and molecular characteristics of the patient's disease. But which kind of tailored medication are in the doctor's bag, please?
Hi. So, okay. This is a picture from an ancient book of rheumatology. Actually, when I started practicing as a physician at the Burlo Hospital, I saw, unfortunately, some children with the juvenile idiopathic arthritis, that is a form of rheumatoid arthritis, who developed such a disabling condition. At that time, one of the medication available for this kind of children was gold salts. Even if there is no evidence on the efficacy of these medications, but they were proposed based on an analogy on the use of these medications in tuberculosis. This was quite common in the past century to try discovering medication through such analogies. It took 60 years to understand, to learn that gold salts were ineffective and in rheumatoid arthritis, and at the same time other medications more effective were developed. Methotrexate first, and with the start of the turn of the millennium, anti-cytokines antibodies and medication which represented a change, a crucial change in the play of therapy, as they were based on, at the first time, on the knowledge of molecular mechanisms underlying the disease. It is of some interest that in part the molecular mechanism underlying inflammatory arthritis or other disorders have been understood through the study of monogenic disorders, as monogenic disorders may be considered a model, a simplified model, in which I can discover a single molecule, a single pathway involved in the pathogenesis. And these models are not actually representative of most multifactorial disorders, but have the advantage to give us some hints on extreme conditions. Thus, this is very important to develop medication like anti-cytokines, antibodies, and many other. It's important for me to say that we have always to see this medication on the timeline of the patients and on how the timeline of the life of the patients intersects with the timeline of the changes in the technology, in the medical technology, in the medical knowledge, as many patients will not have a treatment for their condition, will have gold salts. Others may have tailored medications, but in the middle there are many patients that will participate in clinical trials. That is not always easier. Many patients today participate in clinical trials as the medicine move forward very quickly, and as it is quite common that patients with these conditions have to decide, have to check a consent and information to give their consent, and so on. This is a case of a patient with a monogenic disorder with a progressive arthritis that had similar characteristics, similar features like lupus erythematosus systemicus, the arthritis of lupus. However, this boy started developing this kind of arthritis with deformation in his first years of life. The discovery that his disease was due to a different cytokine compared with those of other forms of arthritis. It was due to interference came from the discovery of a particular group of monogenic disorders called interferonopathies. And thanks to this knowledge, he could experience not just in a clinical trial, but in a compassionate use, a medication aimed at targeting such interference. And now we learn from him that this kind of medication can be used also in subgroups of subjects with rheumatoid arthritis, for example, provided that we have biological biomarkers that can help us stratificate patients for this kind of therapies. This is an example. Depending on when you had born, you can have no effective treatment. We can enter clinical trial. You can enter a clinical trial without having any benefit from them, or with a benefit, or we can arrive when there is a medication just for you. Okay. Here is a paradox. This is wonderful for us to see new medication that can cure patients that were not curable before. But there is a real paradox that this, as the greater the health and safety results from such studies, the greater the perception and of needs and the request from people. The people were more satisfied when medicine had just a few medication than now. This is really a paradox. More I have as a result of technology, more is the request from people. And this is risky. We saw this in the recent epidemics, as this can induce a distrust towards medicine, towards science. And I like to give you these images, this image of the poem from Leopardi about the need to have to think about our positional role in the nature and to balance this also in communication. The risk is that the technology is always more difficult to communicate, to tell to people, and for this reason, many people may be fascinated by the simple idea, the fine idea of a skilled physician who discover the easy to communicate medication to cure some disorder. We saw this problem about hydroxychloroquine in COVID, mesenchymal cells in muscular atrophy, and so on. Thus, my conclusion is that we have to go for this challenge of communicating the importance, the time, the efforts needed to go in the next future of therapeutics. Thank you.
Thank you. And just a question, but what usually do patients ask their doctors about personalized medicine?
Okay, the patients don't ask for personalized medicine as so. They ask for a treatment that can give them relief. And if you don't have such kind of treatment, they can be frustrated by the contrast from a communication that we use in science to tell a journal, a newspaper every day that we reached a great success. And why not for my son?
Okay. Well, is it correct if I say that you see a world where doctors will eventually have a toolbox of different treatments that can be targeted to an individual patient according to their specific genetic makeup? But what are the main challenges to face? I mean, what about the cost? One problem with personalized medicine is the cost. It's a question for all of you. Please.
I can start. I think we have to be clear that patients, and of course there are differences in cultural interactions between patients and doctors. A patient usually trusts the doctor and doesn't challenge the doctor for the recommendations a doctor makes at the time of a prescription and diagnosis. You're right, and I think what's happening is that doctors need to be in continuous learning. That's another problem. We have experience. Oh, I have used this treatment 20 times, I'm going to rely on my experience, my prior experience. And sometimes what we are seeing is that the need to catch up with the evolving understanding of disease and the options that are then available sometimes is not happening because we are overloaded on our daily life as clinicians. So I think this is a reality we have to accept. So not all clinicians are close to science to catch up on those developments. So we have experts, we have specialty physicians, but the GP, the general practitioner, is not always accessing that understanding. Not because they don't want, it's a reality of life. But the assessment of cost and benefit, and that's why I think in the UK this is done very clearly, the recommendation of what is effective and cost effective as well needs to be done by an independent body. It's not the doctor who should decide. There should be a consensus from experts that says, okay, this drug has a positive benefit risk, which has been approved by regulators like the EMA, but then is it cost effective is a separate question that is not part of the approval. And this is something that unfortunately has not been happening systematically. Some groups take the opinion of a group of physicians or a certain association, and that's not always a consensus.
Paolo Vicini.
Maybe I can comment. Maybe
there is a perception that all these new science, computer, simulation methodologies are making things more complicated, but that's actually not what's happening. We're trying to make them simpler. We're trying to understand a little better what happens, because empiricism only goes so far. And the magic bullet, that Alberto was talking about, while it sounds attractive, the expert that knows everything and can recommend the right molecule just so, is increasingly difficult, because we understand a lot more about disease mechanisms, we understand a lot more about what needs to be done, and maybe there's a couple of things that we can do to improve the situation. One is we need to be better, I think, at identifying the cases and the examples where this type of personalized approach actually makes a difference. So we want to know which patient groups can benefit the most from the investment that may be needed to develop personalized medicines. And the other thing that I think that we need to get better at is to percolate down, I think like both Oscar and Alberto were saying, to percolate down the results of the science that we work on to clinical practice. And there's a number of reasons why that doesn't happen. Maybe the science is not mature, maybe the examples are not quite there, the strength of evidence is not quite there. It's an ongoing process. But I think the more the patient can directly benefit from these advances, the better off we're going to be.
Marianna Lucafò.
Yeah, thank you. From a laboratoristic point of view, and probably one of the most important challenges related to the standardization of the protocol, for example, used to obtain results useful then for personalized medicine. For instance, research about organoids and the use of patient-derived organoids for predicting drug response in cancer patients. There are problems related to the protocols used in different laboratories. We cannot merge all the results. And this is a big problem, because we can obtain a final solution, because we need more data. Okay, so this is a problem. Then, of course, regarding the use of patient-derived organoids for predicting drug response, we need to evaluate the clinical value. We are in a preliminary step, and for example, we are waiting for the results from randomized clinical trials in which we will see how the patient outcome could be different, comparing the physician's guided standard of care treatment compared to the SA guided treatment using patient-derived organoid drug screening. Patients receiving a treatment based on the use of patient-derived organoids drug screening should benefit clinically, and this benefit should be also cost-effective with respect to the standard care treatment.
Thanks. I have more questions for you, but hands up if you have a question for the panelists. Do you have a microphone?
I was thinking previously, since you mentioned that it takes many years from the preclinical studies till the approval of the drug, I wanted to know how come the COVID vaccines were approved so quickly, and how can I be sure that in 5 years from now or 10 years I won't have side effects?
Is the question for...
May I start? May I start to answer your question? I think it's a very important point. Remember, evolution is happening all the time, and fortunately, I would say fortunately, the mRNA technology was already being developed for other conditions, but it's a platform which you can use in multiple diseases. So why were vaccines able to achieve evaluation faster than usual? Because the platform was already there. Not for COVID, of course, but it's a platform. So it's a set of tools that can be used. And I think that has expedited the fact. And remember, people realized there was need to prioritize. There was a huge effort to stop other activities, to put resources, personnel, qualified personnel, to invest in that area because it was an emergency. So I think it's very important. It was not something done without planning, without thinking. There was a platform and there was resource being reallocated to prioritize an area that was perhaps being developed for a completely different disease. Yeah? And Paolo, I think you can.
Yeah, just that's exactly right. Maybe the only thing that I would add is that the reason why these processes take so long is that normally we work on several drugs at a given time, every company does, and so the effort is never focused on one end like it was with the COVID vaccines. The regulations and the steps that I have very briefly described were put in place after some tragedies which I'm sure everybody knows that happened in the last century, right? Then the drug has become a lot more, the drug business, the drug discovery and development business has become a lot more regulated, and rightly so. And while I wasn't involved in the COVID vaccination process, I, of course, like most of you, have read extensively about it, and I think that when it comes to the steps that are required for the regulation of new medicines, including vaccines, none of those were skipped. They were done more rapidly because of the resource investment that Oscar has mentioned, which was unique in the history of humanity, I think. But the process was the same, as far as I know.
Alberto Tommasini, Marianna Lucafò, if you want to reply, or one more question?
Thank you. Okay, first I want to say that the personal medicine is a great advance for the humanity. Secondly, given that the model using in machine learning and the AIs are trained already with the information that we have, and given that the information, the whole genome sequence and the genomics in general is constructed with the data of the population, most of them the European Union or Americans, there is a great bias in the information that we are using to train the models that we are trying to apply to the whole world. So how can the science make front to this bias in this model? Because in this way, the personal medicine is just applied to this kind of population.
You start. So should I start here? Because I think I have published a paper regarding trial diversity this year. It was published early this year. We have to be careful about diversity. We are different, but we are still Homo sapiens. Yeah? So we cannot start from the differences to discover drugs. We need to start from what's common and understand whether the differences are clinically relevant. And in some cases, they are. In some cases, they are not. Actually, in many cases, they are not. We are all the same. We are Homo sapiens. What is important, I think, is clearly that when there are differences that are clinically relevant, we need to understand whether we can find ways of identifying them. But that clinical relevance is a phenotypical relevance, it's not necessarily a genetic one. And when we say, oh, I have population A, population B, Chinese, we have to study Chinese. How many subpopulations in China there are? You have 51 ethnic groups, but probably thousands of genetic differences in there, as many as genetic differences in Africa. So I think when you call someone a white, a black, a Chinese, a European or not, we are really biased culturally. This is not what we're talking about here. We're talking about understanding how phenotypical factors, genetic factors, but also lifestyle matter for the outcome of a treatment. So we need to be absolutely clear. Genetics is one component, an important one, but it's not alone. And the way we look at the phenotype sometimes is not really what matters for how disease progresses or for how the treatment performs. And that's why, just to close, just what I think is important, because we cannot find patients and test patients forever. That's why the virtual element is important. We can do something we call what-if scenarios. What if the genotype was different? What if the organ function was different? And I can do that in the computer. That's why what the power of the virtual twins are. I can now create patients that I haven't been able to access. Rare diseases. How do I make patients with rare disease? They don't exist. I have a treatment now with 8 patients in a trial, and that's all across the world. How do I know if the drug is going to work in the future? I know the evidence from these 8 patients, but these are rare conditions. How do I get confidence? Through virtual twins. I think the solution is, of course, there's a bias. I completely agree with you. Sometimes the information is not coming in the mainstream, but we need to rely on the technique to help us overcoming the paucity of data. That is not always easy to get.
C'è una domanda a Lilla.
Thank you. Yeah, very interesting discussion. Pharma companies are developing drugs and selling that. For precision medicine, you need algorithms that are medical devices, by European rules that define if you have to use the drug from company A, B, or C. Who do you think in the future is going to develop this algorithm? Because a company probably has a bit of problem to propose other products. So this is a challenge that will be significant. No?
Paolo?
I fully understand the question, but I think what is being asked is whether the development of certain devices for personalized medicine will rely...
Yeah, the algorithm, in general, they are under MDR, so they define as medical devices. In the US, CDRH are approving them, also based on AI and so on. So the point is, who is going in the future industry of personalized medicine to develop and sell this algorithm that need to be integrated in the hospital settings, define if you're going to use the drug from GSK, Pfizer, or Merck, or others, because it's a bit of it's a tricky problem.
So I think, so this has been, you're right, I mean, if you're going to use something in the process of diagnosis or prescription, you need device status from the EMA. It's true, and also from the FDA nowadays. So I believe this is really a gap at the moment, that industry is not taking the responsibility in general. But you're right, I mean, who is going to take, and perhaps some of you might be sufficiently knowledgeable in the technology, the informatic, the IT technology that you need to build together with these algorithms to help physicians make decisions in an independent manner from a specific product or source. And I think this is a gap indeed in the evolution of this process, because we do need those devices, like we need devices for other types of conditions. But I'm not aware of, I know of one company that is trying to do this. They are based in New Zealand. It's called RightX, basically the right prescription. This is developed by university, and they are using in a hospital setting, specific for the hospital in New Zealand. That's the only one I'm aware at the moment. I don't know if you know of other.
Yes, I know that there are many candidates just now, for example, to read X-ray that are fine, and may be that they are already better than many radiologists, but there is a discussion about how to authorize these devices just like drugs, but just like drugs, the responsibility of the final use will be to the physician. Yeah. And just like you can read on the most authoritative books, these are the best knowledge. These are, but are not a rule to use on a given patient. Are just an advice.
An advice, yeah.
Well, the last question, please, be short.
Thank you. Just a quick question, I think. When talking about tailored medicine in the future, what do you think should be the focus? In developing new drugs that serve as a panacea for everyone, or in developing effective biomarkers and then recurring to those drugs that are already discovered?
So I am probably both. I don't know. Regarding the predictive biomarkers, I think that if we can use the right model, which mimic, okay, perfectly, probably no, but it's very close to the disease, that kind of model can help us to identify some good biomarkers. Up to now, we have used several models, such as mouse and I don't know, for example, some nematodes or other kind of models. But the physiology and both in physiology and the genetical characteristics of our patient were not present in that model. So we have collected several data and results that probably they are not so useful to identify a good predictive biomarkers. Fortunately, now we have the possibility to use these new patient-derived models that could be, we hope that will help us to identify new biomarkers. But we need to, these systems are not so complex like an organ. For example, I showed you this intestinal organ, but as you know, our organ can mimic the intestinal epithelium of our intestine. So there are enterocytes, goblet cells, Paneth cells, but that's it. I don't have immune system in that culture. Okay, so we need to improve our models to mimic exactly in vitro, of course, the characteristics of the disease, to try to identify new biomarkers. But of course, it will be for us. It's important then to translate this kind of results by using patients' samples to confirm, to validate our data derived from the in vitro analysis, because we need to confirm the clinical value of our biomarkers. But yeah, we think that in the future this new platform can help our patients and the clinicians during the guiding of the choice of the drug.
Another development might be to rely on combination therapies with medication and doses of medication whose effect is not predictable by human mind, but could be optimized by artificial intelligence algorithms.
So perhaps just to complement, I guess the example from what you're raising here, let's take oncology. Why is the intervention oncology improved so much over the last 2 decades? Because before you treat lung cancer, you didn't treat, you decide on the organ, you have stomach cancer, lung cancer. Now we are talking about mutations, and the diagnostic process has changed from an organ-based diagnosis to a mutation-based diagnosis, and that allowed us to then link the drug to its target. Unfortunately, that same mindset that evolved in oncology has not occurred across many other therapeutic areas, where now I am defining the disease, the condition, based on its real pathology and targeting it. And this is what I think would then define the role of certain biomarkers, as long as we know the pathway. And I think this is happening across other therapeutic areas, including rheumatoid arthritis, where you're learning the pathology, but as we know, resources are needed to speed those things, and certain areas are neglected because there are few patients and there's interests in other therapeutic areas, societal interests, sometimes financial interests, but it's a reality, so we cannot tackle all the problems at the same time. But I think across all therapeutic areas there is this movement towards understanding the pathology, and that's where the biomarkers will play a role.
Well, our time is almost over. The last question is for all of you, be short. What is the takeaway message? I mean, it's hard not to get excited about a future guided by this medical revolution. Your takeaway message for us?
Just start and make it very brief. I hope that we were able to communicate some of the excitement that we have when we work in this area, in this business, in this scientific endeavor, and I hope that we motivate you to learn more about it.
Oscar Della Pasqua.
So I guess I am biased, of course, because I believe that the integration of knowledge and not only the experimental evidence is what's going to make the difference. We are now making decisions on integrated data and exploring the interactions that are not only in one level but in multiple levels. And I think models, mathematics and statistics, no matter how you dislike that, will be playing a role in the future.
Alberto Tommasini?
We have to be very good in explaining both the limitation of our achievements and the solidity of the technology and the science underpinning them.
Marianna Lucafò.
Yeah, I think that among all colleagues and laboratories, we should communicate more to favor the standardization of our protocols, because we need to collect more data, yes, but we need to do this in less time and to collect new protocol for the good, the better patient-derived models, because I think that this kind of patient-derived model will be really, really useful for our patients. So we should communicate more among each other. Yeah.
Well, thank you, and now our time is over. Evolution is happening all the time, you said. Well, let's hope, we trust in your job. Thank you for your contribution, and thank you for your attention. Bye, and enjoy Trieste Next.